have class coming up too. The actual
Hey everybody, I'm Alan Pan. I'm the co-founder at STEM for Tomorrow. I'm a rising senior at James Madison High School, and today I'll be talking about cell life and in-depth look into intercellular cell mechanisms. So today we're going to be going over cell reproduction, exploring mitosis and meiosis, cell signaling, the key to cell communication, cell transport systems, intercellular and intracellular transport, and finally we're going to be going over everything in a conclusion. So first we're going to talk about cell reproduction. So the cell cycle is a series of events that takes place in a cell that cause it to divide into two daughter cells. The eukaryotic cell cycle consists of two main parts, the interphase and the meiotic stage. Interphase causes the cell to grow, accumulate nutrients needed for mitosis, and replicate its DNA and some of its organelles. During the mitotic phase, the replicated chromosomes, organelles, and cytoplasm separate into two new daughter cells. And there are different checkpoints that exist at the different phases of the cell cycle, which I'll go into detail in later slides to ensure proper replication without mistakes. So first in the cell cycle is interphase. Interphase takes place in a new cell's nucleus, which is pretty much the part of the cell that stores all its genetic information. Interphase acts as a preparatory phase for mitosis new cells. And this is the longest part of the cell cycle because it takes roughly around 90% of the cycle, as you can see in the graphic. Interphase is made up of three parts that all focus on making sure the cell has everything in order for mitosis to properly go through the S phase, G2 phase, and mitotic phase. So as you can see here, interphase pretty much is most of the cell cycle, which is the gray part. Interphase part 1, the G1 phase. The cell focuses only on increasing its supply of proteins, the number of cell organelles, and cell size. There's a checkpoint during the G1 phase. It's known as a restrictive phase, directly before the cell moves to the next phase, which is the S phase. And during this restrictive phase, or same thing as a checkpoint, a cell can decide either to continue the cell cycle regularly and enter the S phase. That's if all the different organelles, the supply of proteins, everything is in check and looks good to go. It can go to the G0 phase, which is the phase before interphase, where pretty much the cell's not doing anything. It's not dividing or going through the cell cycle, just put in a permanent state of neutralness. Or finally, the cell can stop during the G1 phase and go back to G0 or just to continue onwards through the cell cycle. So as you can see, like this checkpoint pretty much checks to make sure if everything's all right to continue on the cell cycle to prevent any errors or misshapen cells. Interphase part 2 is the S phase, and the S phase deals a lot with the DNA's replication and synthesis. So pretty much it's creating new genetic information in order to give these cells, while the phase before focused on the number of proteins, organelles, etc. The ending of the S phase has all the chromosomes replicated for new cells, with each chromosome having both sister chromatids. And sister chromatids are each pair of the lines that you see inside the graphic on the right hand side. As you can see, the numbers of blue and red chromatids have doubled. And finally, RNA transcription and protein synthesis don't occur in this stage. For the third part, we have the G2 phase. And the G2 phase focuses on protein synthesis and rapid cell growth for final preparations before mitosis. So this is the final step of interphase. The second and final checkpoint in interphase takes place as the G2 checkpoint before proceeding to the mitotic stage. So pretty much a final checkup. And the checkpoints check if the DNA is damaged and decides whether to repair or commit apoptosis, which means just program cell death. So, like the first checkpoint, it takes care and checks to make sure it has all the different parts needed for it. But the G2 checkpoint makes sure if it has all the genetic information for the new cells. So, seeing the big picture, this is the entire cell cycle, and you can see the different phases labeled as G1, S, G2, and M is mitotic phase. And you can see here, they didn't really draw it in the diagram, but right here is where the restricted phase slash G1 checkpoint is. It's pointing to right at the end of the G1 and is moving in a a clockwise motion. Yeah, and it also gives you what's good about this diagram. It tells you the amount of time that a cell typically takes place in. So you can see that phase takes like 6 to 8 hours, while the mitotic phase is really like the quickest part besides interphase, and it's only 1 hour. So most mammal cells generally take around a whole day or 24 hours to complete division and growth. But with smaller organisms with less different variety of cells, it can take a lot faster. All right, next we're going to talk about cell reproduction through mitosis. Mitosis is a part of the cell cycle when chromosomes are separated into two new nuclei. Mitosis is a process exclusive to eukaryotic cells, and pairs of chromosomes condense and attach microtubules that force sister chromatids to opposite sides of the cell. And the stages of mitosis are prophase, metaphase, anaphase, telophase, and cytokinesis. Telophase and cytokinesis sort of get linked together because that's just the physical separation between the two cells, and everything's already split and everything. So, uh, the video on the next slide will break it down even more, but you can kind of see how mitosis is through all the steps pictured below in the diagram. So I'll just give you a second to look at that. All right, so this is the video I was talking about. It really gives like a good overall understanding on what mitosis is.
We looked at the general process of mitosis in this video. In this video, we are going to add in some extra detail, including the names of the key stages. You may want to watch this video first to refresh your memory of how mitosis works. You should already know that mitosis is the process of cell division that produces identical copies of cells and is involved in growth, cell repair, and asexual reproduction. When cells divide by mitosis, the number of cells increases, and hence the organism grows. Different organisms have different numbers of chromosomes. To keep it simple, we are just going to look at what happens to one pair of chromosomes during cell division. Before we start, just a quick reminder that a chromosome is made up of two chromatids, and one from the mother and one from the father. So mitosis, with our special sciencey names, add in, to this day, I just remember IPMAP, to remember which order the stages go in. Then just tag cytokinesis into the end. Interphase, the chromosomes duplicate and become two identical chromatids, joined at the centromere. So in humans, it has gone from the original 46 to 92. In prophase, in the nucleus, the chromosomes condense, and in the cytoplasm, spindle fibers form. Metaphase, the nuclear membrane breaks apart. The spindle fibers attach to the chromosomes, and the chromosomes line up at the equator of the cell. Anaphase, the spindle fibers shorten and the centromere divides, so that each chromosome becomes two separate chromatids. Telophase, the nuclear membrane forms around each set of chromosomes. Chromosomes spread back out in their new nucleus, and the spindle fibers break down. In humans, each nucleus has the normal 46 chromosomes again. Cytokinesis. The cell membrane pinches into two separate sets of chromatids into two identical daughter cells with the same number of chromosomes as the parent. So 46 or 23 pairs in humans. So there we have how cells divide by mitosis to form two new identical cells with the special IPMAT, sciencey words included.
All right, so hopefully that gave you guys a good understanding about what mitosis is, or just a pretty rough idea of what it is. So next we're going to be talking about cell signaling. What is cell signaling? Cell signaling involves any communication process that maintains a cell's life and maintains multi-cell actions. Cell signaling functionality is important in tissue repair, homeostasis, development, and immunity to disease. Thus, a failure of cell signaling could have really grave results uh, in diseases such as like cancer and diabetes. That all comes from failure of cell signaling. And scientists and clinicians heavily study cell signaling in order to better combat diseases and understand them. So this is just an overview of cell signaling. For extracellular signaling, which is signaling outside of a cell from one to another, a signal molecule will be received by another cell and respond correspondingly through programmed responses. And the typical process for extracellular signaling involves first the synthesis and release of a signaling molecule, then the transport of the signal back to the target cell, then the signal binds to a specific receptor, causing activation. This causes the start of the signal transduction pathways, and the response takes place through cellular response. Uh, and signaling differentiates by distance that the signal has to travel in order to reach the target. Like to clarify on the last point, what it means is if the signal is really far away and it only like sends through like really quick signals, as we're going to be talking about, this is more like muscle cells. It's like an electrical signal, so it doesn't send out that much information. Only like creates a quick response, like a jerk of a muscle. The graphic on the right shows you the pretty, like, one of the most basic uh, diagrams about how overall cell signaling works. So the first type of cell signaling we're going to be talking about is autocrine, and autocrine involves signaling with only one cell. It targets itself by releasing a signal that binds to an appropriate ligand, which it's a fancy word for receptor on its own surface, and normally occurs during the earlier stages of an organism's life in order to ensure the cells are developing normal tissues and have proper function. Autocrine is useful in sending pain and inflammatory responses in organisms to maintain homeostasis. Or homeostasis is just a process of like maintaining body temperature, like sweat, anything to just like keep the body in check and normal. Another example of autocrine signaling is when a cell is infected with a virus. The cell sends itself a signal to commit apoptosis or cell death, which also kills a virus. So that's is very effective as a signal. And the diagram on the bottom shows you exactly how the signals go into the receptors on autocrine signaling. Cell signaling paracrine. So paracrine signaling involves sending signals between cells that are close together, but still two different cells. These signals usually only create a response that lasts a short amount of time, and these signals last a short amount of time because it is removed by neighboring cells or degraded by special enzymes. Like I said, these are like the quick signals that only transmit only a little bit of information and last only a little bit amount of time. And a good example of paracrine signaling is with nerve cells. So nerve cells send out fast electric impulses, and these impulses causes releases of ligands called neurotransmitters that move between closed nerve cells, called chemical synapses. Thus, they move fast and cause an immediate response due to close proximity. So you can sort of think it like a lightning bolt just shooting out from one cell to another, and it just commits one really quick action as the response. So this diagram shows you exactly how synaptic signals work. Uh, you don't really have to get deep into the details about the different parts, but this is the general diagram about. You can see that the blue balls at the top, those are the signals, and the little blue ovals with a circle cut out of it, that is a receptor. So this video kind of goes in depth into how exactly it works.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. The synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
Okay, so just a word on the last video. It's okay if you don't understand every single part of it. Just the main broad idea that I was trying to get across is how the receptors come in contact with the signals and how the signals are spread over only a small distance. So the next type of cell signaling is endocrine. So endocrine signals are like paracrine, except the distance in between the cells are just a little bit longer. And all endocrine signals originate from endocrine cells, which are located in endocrine glands, such as a thyroid gland and pituitary gland. So what's different about the endocrine system compared to the paracrine is because paracrine has, like I said, a quick and immediate response. So in contrary sense, these signals are a lot slower moving and they're carried by the bloodstream, which has a slower response time. It causes a longer lasting effect. And a good example of endocrine signaling is hormones. And hormones travel long distances through the bloodstream at a relatively slow transport. So, as you can see, these cells are a little bit more than a long distance away from each other, and it has to flow through the bloodstream in order to reach it. So, these cells can be like in completely different parts of the body. The next type of cell signaling are gap junctions. And there are a lot of gap junctions in animals and plants, and there are connections between the plasma membranes of neighboring cells. And the connections are water-filled channels that allow small signal molecules called intracellular mediators to diffuse between two different cells. So think about how a cell has a plasma membrane that filters out what comes into it and what goes out of it. And in the plasma membrane, there are just channels filled with water that allows different ions of different elements to just flow between two cells, which require no energy or not a lot of movement anyways. So, like I was saying, small ions like calcium are able to move between the membrane, but it blocks out larger molecules like DNA and proteins that can't fit through. I like to think of it like sort of like a mesh that only filters out small particles and keeps the largest one back through it. And due to the restrictiveness of these channels, cells are able to easily transmit signals. And a good example of gap junctions is in the heart muscle, where electrical currents are generated by the transport of inorganic salts. So the electrical signals, that's more of a paracrine, are like a really quick signal, like electricity. And but the paracrine signal is caused by the transport of these salts, these small ions, through the gap junctions of cells. So intracellular transport, it can like be tied into many different ways. Like one can cause the other. It's just a signal and a receptor. So this is a diagram of a heart muscle, so you can get a better idea of exactly how gap junctions are playing a role. These two orange or like flesh-colored blobs, they're each a cell at heart muscle cells, and you can see that the gap junctions, which are like the gray line, normally is a single cell's membrane. So you can see that there are little parts of it which are brown that allow the gap junctions and allows the calcium ions to flow about. And also you can see the electric currents in each cell too. So next I'll be talking about cell transport systems, exploring intracellular and intercellular transport. So the graphic on the right gives a basic idea about what we're going to be learning about passive transport, which you can see it flows from high concentrations to low concentrations to fill up and be balanced. And active transport, you can see ATP in the spiky box, which means the ATP is an energy source that is used in active. So that just gives you a little sneak peek about what two transport systems are. So what is cell transport? Cell transport is the basic process of moving things in and out of a cell. The two different categories of cell transport are passive and active transport. Like I said before, the difference between the categories is how much energy is needed to move the substances in and out of the cells. So active needs energy, while passive does not require energy. And the next slides will break down exactly how, what are the two different categories and the different types in each. So this shows you the structure of the cell membrane. I kind of went into that with the last diagram. You can see that it blocks things and lets other things through. So passive transport is the movement of atomic and molecular substances. Think just like small particles like calcium ions or any kind of small ions or water, things like that, across the plasma membrane into the cell without any energy input. So the three types of passive transport that I'll be explaining on, and which are mainly focused in biology, are diffusion, facilitated diffusion, and osmosis. So the first type of passive movement would be diffusion, which is the movement of a substance from high concentration across a concentration gradient, the difference in the of concentration to a lower concentration. So pretty much what the concentration gradient is the separator or barrier between high concentrations of one molecule versus a lower concentration on the other side. So for this example, the concentration gradient would be the lipid bilayer or the cell membrane. It's the same thing. And you can see that the particles slowly move across until the concentrations are just about balanced. Then there won't be any more movement. And that's the whole point of diffusion, to just get it equal concentrations of any kind of substance. So next is facilitated diffusion. And facilitated diffusion involves the movement of molecules across the cell membrane through special transport proteins embedded in the membrane. The protein channels allow water molecules and small ions to pass through the membrane without touching the hydrophobic tails of the membrane. So this is just for like larger proteins and things. It doesn't take any energy, but it just goes through a carrier protein into the other side without like letting water in and things like that. The carrier proteins are a type of channel that binds with ions and molecules, causes change in the shape of the proteins, thus carrying the ions across. So yeah, what carrier proteins do, it takes in the ion and it holds it and then just lets it out the other side because of how the ion alters the shape of the protein. The third type of passive movement would be osmosis. And osmosis is a specific form of diffusion in that it only involves water molecules. So water passes from high concentration regions through a semi-permeable membrane to low water concentration. And the semi-permeable membranes are thin layers of material bordering a cell that allow some things to easily pass through, but also prevents other things. So like we were saying, the cell membrane is a semi-permeable membrane because of the filtering aspect that it has. So for example, the plasma membrane of cells allows oxygen and water to flow through, while it limits larger molecules like glucose and protein. And you can see that you can see the solid molecules inside the water and how it passes through, just through the water. Next, we're going to be talking about active transport. And what I kind of talked about before is that active transport involves the use of ATP, which is adenine triphosphate, or 3-phosphate ions. And what I explained in my last video as well is ATP is broken down into energy by releasing one phosphate ion from the triphosphate. So it makes adenine diphosphate, which means 2-phosphate, and releases 1-phosphate as energies. And it uses this energy source to move substances from a low concentration to a high concentration of a given substance. So it does the opposite of what passive transport does, which is like the balancing of high concentration to low concentration. So you're gonna have to spend a little energy if you want to do the opposite. All right, that's my entire presentation. Thank you. If anyone has any questions, please leave them in the chat and I'll answer them for a little bit. And if you guys don't have questions, you're free to leave.